Hi, everyone. Thanks for having me here. And we are going to change the gears a little bit from traditional tall corn talk to something that's like a new potential going forward and something that has a lot of unanswered questions. And we are here to brainstorm and answer those things. So, today, I'll just start off with explaining a little bit about what is short corn, how it came into being, and what are the agronomic considerations when we look at it for silage purposes. And secondly, how the preliminary trials that we are doing at UW-Madison in collaboration with the industry fair in those considerations. And eventually we'll talk about some challenges and some unanswered questions that we'll be focusing on going forward so that we have something which is an alternative to our current systems and is also easier to manage. So when we look at short corn, it is, as we all know, genetically and selectively bred and is shorter than our traditional corn plants. And the shortening of the corn is happening because of the smaller internodes. So we have smaller distance between two leaves as opposed to the traditional corn. But another thing that we see in our shorter corn plants is that the ear height is closer to ground as opposed to what we see in terms of our traditional corn plants. But when we grow short corn in the field, about what time those differences are actually becoming visible is somewhere around the vegetative state V10. That's where the differentiations start becoming more vibrant, and we can tell which plots are what going forward from that point. So while the breeders and scientists were trying to look for alternatives to the problems that we have with tall corn, a lot of inspiration for developing short corn hybrids came from dwarf and semi-dwarf varieties of rice and wheat. So, in order to do that, we were looking at different pathway genes that were responsible for corn growth and elongation. So, the three primary pathway genes that were affecting the corn growth were the gibberellin pathways, the BR pathways, and the auxin pathways. All these are crucial for different types of growth in the plant. For example, the GA pathways, they are responsible for stem elongation. The BR pathways are responsible for general development and regulation of growth hormones. And then the auxins are responsible for cell elongations. So now the breeders were looking for something which can actually uh, altering post impression can actually shorten the plant, but also not affect our overall yield and reproductive health of the plant. So there were two gene families that were identified, which were very similar to the dwarfing genes that were seen in rice. And these could eventually be altered in a way where we could alter our plant by affecting the vegetative part without any impact on our overall reproductive part of the plant, so that we were still preserving the ear strength, the ear size without impacting by reducing the overall height of the plant without impacting the yield. So when we are looking at the modification of a corn plant like this, we are also very much interested in understanding what are the agronomic considerations that are required or that are actually being answered because of these modifications. So just to start off on that, the first agronomic benefit that we want and we are getting from short corn is the improved standability, and that is by the resistance to lodging. Here I have a picture of the Derecho storm from 2020 and a quick analysis of lodging studies across the upper Midwest also show that at different stages of at different stages of corn growth, we observe different losses in terms of yield. In terms of yield. For example, during the vegetative states from V13 to V15, the losses are somewhere between 15 to 30% for the plants that kind of lodge around pollination, etc. The losses can go as high as up to 50%. And in the grain fill, they can be capped somewhere at 25%. So now, if you remember from the picture that I showed you a few slides ago, the differentiation of short corn and the tall corn kind of begins around B10. So which means that the stages where the lodging potential or the lodging damage would actually impact the plant uh, is where the differentiation has already occurred and the plant can actually be preserved from that damage. Going forward, we are also looking at higher nutrient and water use efficiency because now we have a plant which has different architecture and would probably have different distribution of these resources in it and would then eventually have a higher efficiency or a lower efficiency. That's something we are answering through our research going forward. And the last one is the easy accessibility for different operations, which may consist of fungicide applications, insecticide applications. So going in the field with the equipment is easier because your corn is about 1/3 to 1/2 shorter than what we are traditionally used to. So these agronomic considerations are actually ubiquitous for both grain and silage. So specifically, when we look at silage, there are other considerations as Dr. Geyser were pointing out, and most important of them are the yield, the digestibility, the nutritional composition, and energy content. So if you see at the figure on the slide, uh, if we divide the different types of hybrids into 4 quadrants, what we want is our hybrids to lie in the 4th quadrant. That's the sweet spot where we have high tonnage, we have high milk yields as well. Uh, if our hybrids are lying somewhere around high tonnage, low quality regions, or uh, uh, low tonnage, high quality regions, those can be compromised. Those are the compromises that can be made suiting to one's needs in if need be. However, where we definitely don't want our silage is this part. So in order to test where our corn hybrids or short corn hybrids at this moment that are available to us fair, we conducted some research in collaboration with the industry, and I'll be showing some preliminary results from that going forward. So if you look at the short corn silage trials that we conducted at Arlington last year, we see that the short corn hybrids had yields which were very comparable to the taller hybrids, and they were and when we split these taller traditional hybrids to the uh, we separate out BMR hybrids from the traditional hybrids, we see that the shorter corn hybrids had higher yields as compared to BMR hybrids as well. Beyond this, when we are all looking into the other considerations of digestibility, the short corn hybrids were actually showing really good digestibility, which were as close to BMR, and it was actually faring really well in comparison to the taller hybrids. So one thing that I would like to really point out here is, although the digestibility that we were seeing was higher in short corn hybrids as compared to the taller corn, there was a lot of variability in the data that we were seeing. So that's actually one of the goals of this kind of research or hybrid testing that we are doing, where we identify the hybrids which are on the lower end of the spectrum, and then we kind of eliminate them going forward so that we do have the hybrids selected which have our uh, are desirable characteristics going forward. Looking at other numbers in terms of predicted milk yields, the shorter hybrids did fare well and fare better than the BMR hybrids. And if we go further and look into the energy content, because as we have heard from earlier speakers, these are the major considerations that go into deciding what are deciding what feed is better for our livestock. We did see that the short corn starch content was higher than the BMR hybrids, but it was actually comparable to the taller hybrids. But I would like to specifically highlight this point over here. The range that we were seeing in the shorter corn hybrids were anywhere between 31% to 37%, which was not true for the taller hybrids. So there was a lot of variability among the hybrids in case of the shorter corn hybrids. So therefore, again, reiterating my previous point, we are in this selection phase where we would be trying to select up for the hybrids which are having more desirable characteristics for our needs. And if I try to plot these relationships, or if I try to visualize what we were looking for our corn silage or silage corn, starting with the considerations and with the desire of ending up in the top quadrant, the preliminary scatter analysis of the data shows that a lot of short corn hybrids were actually faring in that top section of the quadrant. So this means that there are hybrids which have potential to actually perform as well as the tall corn hybrids, and could eventually be a path where a lot of our corn looks different than what it is today. When we are looking at the yield, the quality, and everything, there are so many questions that arise with respect to management conditions and management decisions. So if we look at the research that has been happening around the Midwest, specifically led by industry at this point, we see that for nitrogen responses, there were no major differences between the nitrogen responses for short corn and tall corn when the application was done at V6. And similarly, when it was done at V12, there were not major differences. So if you see these graphs on the screen, the yield delta actually represents the deviation, or the positive delta represents the increase in yield due to additional nitrogen application at the specified stage, and the negative delta shows the hybrids which were performing poorly as opposed to the usual nitrogen application rates. However, there was very interesting observation that was reported by actually this study only, and that was the nitrogen distribution in short stature corn. Although the short stature corn responses were very similar to what we were seeing in the traditional corn with respect to nitrogen, the overall distribution was very different across various stages. So during the mid vegetative stages, we did see that the leaf nitrogen content was 13% higher than the traditional hybrids, and the stock nitrogen content was 18% lower. And similar deviations were observed in terms of flowering and at maturity. Now, all these questions, all these research studies, and all these attempts to answer certain questions lead to further questions where we actually want to see how these distributions would eventually frame the nitrogen recommendations for corn silage and for corn grain going forward. Moreover, we also see that there are folks down in Indiana doing some studies on short corn, and it has been observed from the preliminary data that short corn is actually performing better than tall corn when we are going for higher seeding rates. So in those scenarios, how the nitrogen recommendations change, how the plant populations change, those are the unanswered questions, and that's what we are looking as we are going forward. Further, we are also looking at what are the other potential stresses to watch for when we look at short corn for silage purposes. And most of the diseases and insects that at this moment we think are a threat or are a problem for short corn are very similar to what we see in traditional corn. But I would like to highlight the top three in each category over here. So if we see in terms of diseases, the foliar infections are actually what is of greater concern. And that's because we are seeing that short corn may lead us to go for higher populations if it is performing better in higher populations, but then we would have issues like gray leaf spot, and also going forward with tar spot as well. And then the third point that I would like to highlight over here is southern rust, which is not a greater deal in taller corn as compared it is being expected to be in shorter corn, and that's because the plant architecture is different, so the airflow is quite limited, which may actually provide a breeding ground for a pathogen like southern rust. In terms of insects, there are few insects that are very similar to what we see in tall corn, starting with corn rootworm, western bean cutworm, and which would eventually relate to our problems with ear molds, etc. And then going forward, as Damon and John were mentioning, with mycotoxin risks, etc. Furthermore, we also might have issues with leaf aphids, but all these uh, at this point are, I would say, just potential thoughts and potential diseases and stresses that we are concerned and we are looking to look out for. Uh, Going forward, we are also trying to highlight what are the questions that are required for further testing the suitability of corn silage or of short corn for silage in our systems. And that the number one concern that we are hearing from the growers who have tried putting some short corn uh, in their fields is that their current machinery is not able to pick up the entire thing from the ground because the ear height is quite closer to the ground. Therefore, those modifications with the machinery or modifications with the plant architecture are some things that we need to look forward to. Beyond that, as I mentioned earlier, appropriate or optimum seeding rate scenarios with row spacing, those are the questions we are looking into. One more thing I would like to point out is about the dry down and moisture levels, because these are the key factors which affect how our silage would do in ensiling during the ensiling phase. So currently what we are seeing is with shorter corn, the greenness index or the greenness longevity is higher. So that's one thing we want to estimate and understand, how is the dry down faring with that, and what are the moisture levels at the harvest stages, so that we feed healthier silage into the silos, so that at the feed out we do have a higher quality product. Going forward, we are also concerned about the nitrogen considerations, where we want to see how the distribution eventually informs our decisions, and uh, the short corn applicability or effectivity in the watershed systems and in the alfalfa interseeding systems. And with that, I would just like to highlight the overall take homes and summary from this, where we have definitely seen that short corn is sturdier than the taller hybrids. It is resistant to lodging. Secondly, the yields that we are looking at are comparable to taller corn. And moving forward, the nitrogen responses as tested as of yet were quite similar to tall corn. And with that, we are also speculating that it may or support higher populations than taller corn with the similar resource under the same kind of resources. So with that, I would like to say these are the questions, these are the researches that we are leading forward to actually probably create a next food security revolution in terms of corn. And before I close down, I would like to plug in our corn hybrid testing for 2025. The forms are up on the website. So if you scan this QR code, it will land you to the corn entry forms. And if you're a grower, if you're an industry rep who would like to put in some seed in the corn hybrid evaluation trials, please consider filling up these forms and reaching out to us, and we can move further with that. And with that, I would take any questions that may have come up in the chat.